Hello, tonight's a real short PowerPoint. We're going to talk about the structure of a DNA molecule in a lot of detail. So let's take a look. Okay, we know that a DNA is a double helix molecule made up of nucleotides. Hopefully you remember that part. Okay, the DNA has side chains made up of a deoxyribose sugar, a phosphate group, then a deoxyribose sugar, phosphate group, and so on. Coming off from the deoxyribose sugar, you have a nucleic acid. There are 4 of them in DNA. You could have a guanine, a cytosine, thymine, or an adenine. Okay? Let's go back. Adenine binds to thymine with 2 hydrogen bonds. Guanine binds to cytosine with 3 hydrogen bonds. Okay? Oops, wrong direction. All right, here we go. Now, deoxyribose is a pentose sugar. That means it's got 5 corners. Okay? It has 5 carbons. The carbons are numbered 1, 2, 3, 4, and 5. Oops, 1, 2, 3, 4, 5. Right there. That's how they're numbered. That's important because we're going to talk about 5' and 3'. And why that's important. So we need to know how they're organized. The reason is, in a deoxyribose sugar, on one side you have them labeled 1, 2, 3, 4, 5, with the 5 side pointing up and the 3 side pointing down. This is called the 3' to 5' direction. Okay. On the opposite side, they're upside down, with the 5 pointing down and the 3 on the top. This is the 3' end, and that's the 5' end. Okay. We can only attach nucleotides to the going in the 5' to 3' direction. So a DNA polymerase, which is going to copy DNA, can only work going when the 5 is pointing down. If it's attaching this side with the 5 pointing down and the 3 pointing up, it can only build this way. So when we talk about DNA replication, when we talk about DNA replication, the orientation of the sugars is really important because one side of the DNA will copy just normal. It'll copy going straight up. But the other side, if this is the side I'm building, I can't start at the bottom and build up because this is the 3' end and they can't attach the nucleotides in that direction. So they start here and have to build down. And then they attach back up above that and come down to this one that's already been copied. And then it goes above that one and comes down. And it continues to do that in little short segments. And so the orientation, the structure of the DNA molecule is extremely important when we're talking about protein synthesis and DNA replication. So DNA structure is very important. Okay, let's look at the rungs. You should recognize the rungs just by looking at them. You don't have to memorize that this is a cytosine and this is a thymine. I'm not too worried about that. But which ones are purines and which ones are pyrimidines? Okay, which is which? Adenine, a double ring, will only bind to thymine, a single ring. Guanine, a double ring, will only bind to cytosine. Okay, I already told you that adenine and thymine have 2 hydrogen bonds, cytosine, guanine have 3. The difference in the number of bonds helps to prevent bonding to the wrong molecule. Now, purines have double rings. See, this is just a general shape. Purines have a double ring, so adenine and guanine were purines. The short word is the bigger molecule. The long word is the smaller molecule. Cytosine and thymine and then uracil found in RNA are all pyrimidines. So a nucleotide has deoxyribose sugar, a phosphate group, and one of those nitrogenous bases, one of those nucleic acids. It's the same word, by the way. Okay, some of the differences between DNA and in bacteria and in eukaryotic cells. Do you remember, bacteria are prokaryotic. They don't have a nucleus and they don't have organelles. Eukaryotic cells have a nucleus and they do have organelles. So first of all, DNA in a bacteria is not found in the nucleus because there isn't one, but it is found in the nucleus in a eukaryotic cell. Also, bacterial DNA they only have one, and it's circular. It's not double helic, like in a eukaryotic cell where you might have lots of chromosomes. Bacterial DNA does not have histones, they don't form chromosomes, and they only have a few hundred genes if they're lucky. Eukaryotic cell has histones, they do form chromosomes, and there are thousands and thousands of genes. Okay, so what is a histone? Okay, that's a good question. Histones are proteins that DNA winds around to create the chromosomes. So in a later PowerPoint, we'll go into a little bit more detail about this. But you have your DNA strand. It wraps around a histone molecule and another one and another one and another one. And those then get pulled tighter and tighter. And they get more and more condensed and eventually form the chromosome. So it's a way of winding up, kind of like a spool of yarn or a spool of thread. It makes it a lot easier to deal with when it's all wound up rather than big and bulky. Okay, so what I would like you to do tonight is to draw a, let's see, what do I want you to draw? That's what I want you to do. I want you to draw a DNA molecule. Okay, I want you to have your sugars oriented in the correct locations, and I want you to have at least 10 bases, 10 rungs to your ladder. Okay, with hydrogen bonding in between them. So just make a drawing tonight. Color code it, something like that, make it pretty. Okay, that's it for tonight. Have a great night. See ya.